Okay, welcome back. So this lecture is going to deal with Punnett squares, and we're going to do a monohybrid and a dihybrid cross. Now, they can be tricky, but I'm going to show you the steps on how to do it. And if you go through the same steps, the same order every time, you should be successful. Now, if it's giving you trouble, come to the online class time, or if that doesn't work with you, email me and we can set up a one-on-one session to go through it. I did this with some students in the summer. We would have done this a lot in lab. That's one of the things. We spend like 2 weeks on genetics, and a lot of it's doing Punnett squares. And the best way that you learn how to do them is just to practice. Now, we talked about, genotype and phenotype and Mendel's cross and how he made all these observations. So later, a guy named Punnett, so that's how we get the name, he found the method to how to predict the offspring. And from this method, we were able to actually observe the genotype and phenotype. So it's prediction of what these crosses would be, and then we use that to help support the math and the observations that Mendel observed. So when a Punnett square is, you're going to have the parentals that are going to be on the side, and we're going to separate their alleles into two separate alleles. This is when the alleles split in meiosis. It has to do with the law of segregation when it happens in meiosis, because remember, in the egg and sperm, you only have one version of each allele. So however it splits out has to do with assortment and alignment and segregation. So what's going to happen when you get the other allele from the other parent that gives you your combination? What's going to happen is we're going to split the parents on the top and the bottom, or the top and the side, sorry. And we're going to do a combination of the alleles they donate. And then each of these four squares right here will give us a possible offspring combination. And then we can take the genotype that we get from the different crosses and come up with a ratio for what we're going to observe. So this example right here, we crossed two purple flowers. They're both heterozygous. They have a big B and a little b. And when we did that, we observed that 1/4 of them are big B, half of them are going to be heterozygous, big B, and 1/4 of them are going to be little b. So this tells us both the combination of genotype and phenotype. So how do I do that? As I mentioned, if you do it in a certain order, it makes a lot of sense. It's a little bit of word problem reading. So I'll show you some prompts and I'll walk you through them. First thing is read the problem. Highlight, it's going to tell you which trait is dominant and which is recessive. So you're going to go through this, and I always write out a legend. Even if it's a simple monohybrid, taking the time to do this makes your life a little bit easier in the end. Now you're going to go and figure. I put figure out. So you're going to figure out the genotypes of the parents. You might have to use some word cues. Sometimes it tells you what it physically looks like, and then that will tell you exactly what the genotype is. Sometimes you might have to read. It might tell you it is a heterozygous blank, and then that should tell you it's like, okay, it has dominant phenotype, and it says heterozygous, so that tells what genotype is. Once you figure out the parents, then we're going to do the Punnett square. We're going to have one parent on top, one on the side. And we're going to split our gametes and then fill out the Punnett square. It's really not too difficult. We're going to start with the simple ones first. And then finally, calculate your genotypic and your phenotypic ratios. So if you kind of stick to this order, they're really quite fun to do. I really love doing these. I think they're fun. So we're going to go through one. I'm gonna try to type and write on this as much as possible. So in a certain yellow plant, yellow fruit, we're using the letter A, is dominant to white fruit, which we're going to note with a little a. A heterozygous plant with yellow fruit is crossed with a plant with white fruit. So determine the probable genotypic and phenotypic ratios. So first thing, let's do our legend. So we're looking at fruit color, and it's telling us yellow is dominant to white. So I'm going to type up here my legend. So remember that if it is a dominant, it can have two different possible genotypes. It could be homozygous dominant or heterozygous. So right there is my first one right there. And then let me put a little square right below it. Little y equals white. Okay, so here's my legend. So I know these two combinations of the genotype gives me this phenotype, and this combination gives me this one. Try to pick, oh, I put Y's. Sorry, these should be A's. Let me fix that. The reason that I shouldn't have said that, try to pick letters that look really different from the uppercase and lowercase. Sometimes it's habit. I've used Y a lot. Fix that. So ignore this. My mistake. So here's our letter we're using. As I mentioned, when you do these, especially if you're handwriting it, make sure you pick letters that have a really big distinction from the uppercase and lowercase. So that's why I switched from Y to A, because if you're handwriting it, sometimes your Y is kind of getting that in-between range. So that's it. We read the problem. We found out what's dominant, what's recessive. Now we made our legend. So let's recap. Now find the genotypes of the parents. A heterozygous plant with yellow fruit. So I'm going to write parent 1. They're heterozygous, so that tells me they have both alleles combination. Okay, so there's that one. And if you go, there's parent 1. It's crossed with a plant with white fruit. So using the phenotype right here, you know the only way that it could be white is if it has this genotype. So then we're going to put parent 2 equals A. So now what you're going to do is you're going to take each of these parents and we're going to move them to the Punnett square. One parent we're going to move to the top, and one parent we're going to move to the side. Now, thanks to meiosis, what's going to happen, these alleles are going to split. So for parent 1, we're going to have a big A here and a little a here. For parent 2, we're going to have a little a, little a. All right, so if you look at that, what I did was I split the alleles. This is thanks to meiosis, all right? We're going to separate them, and then boom, right? So this is this egg, this is it, this egg, maybe this is this sperm, this is sperm, and this is another sperm, all right? We have to separate the alleles. You only get one of each allele when it comes to meiosis. Now what you're going to do now is fill out Punnett square. Now how you're going to do this is you're going to bring this allele down to this box, and then this allele across to this box. So the combination right here will be big A, little a. Right, and then you're going to do the same for this one. So this allele comes down here, and then this one goes across right here. So this one will be little a, little a. And then you're going to repeat the same for each of the boxes. My recommendation, always keep the uppercase first. It makes it a lot easier when it comes to counting up everything later on. So let me fill in this one right here, big A, little a, and then this last one, little a, little a. Okay, last one, we got that done. See, once you do that, you're good. So now we're going to determine the genotypic and phenotypic ratio. So I'm going to do that over here on the side right here. So genotypic, all right. I'm just going to write geno. You can write geno or pheno, makes it easier. So what you're going to do in the geno is you're going to count the genetic combinations. So what I would do is right here, here's our first combination. Sorry, A. What I do is I always start in this first square right here. And then I write down what it is. It's a little a. And then I count how many fall into there. And then what I do is I check it off. So if I go through, I see big A equals 2. Right? And then sometimes one thing I like to do is, you know, if I counted it, I put a check mark so I know that I count it and I don't count it again. And then I go to my next one So I've checked those two, and then I have this combination right here, and I count them. So right here, little a equals 2. And then I'll go back, check. All right, did I count them? Boom, 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 right here. Now, one way to check to make sure you have everything, especially when you get to the dihybrids, this number combination should add up to 4 because you have, let's switch the pointer, 1, 2, 3, 4. That's one way to check. So one thing you would type, your genotypic ratio then is this combination, 2 to 2. So you do like this, or you can reduce it down to 1 to 1. So this is your genotypic ratio. So it's just combination. You don't have to reduce. If you want to leave them at 2s, you can. So half of the time they're going to have big A, little a. They're going to be heterozygous. Half the time they're going to be homozygous recessive. So now let's move on to your phenotypic. So this is where I use this to help you determine what it is. So what you can do right next to it is probably write down. So if you look at this combination right here, sometimes I type it right next to it. So the phenotypic ratio, when I this combination right here, I go to my legend. So big A, little a means it's yellow. So let me switch right here. That means these guys are going to be yellow. And then I'll go to the next one. Little a, I know those are going to be white. Okay. Now, the phenotypic ratio is you match up the combination. So with this one, yellow are 2 and 2 white. So then you will just type it, same thing down here. Phenotypic ratio is 2 to 2, or you reduce it down to 1 to 1. Now, the next one should be a good example. These won't always be the same. Sometimes they will be different depending on the combination you have right here. But this is just kind of walking you through. So let's do another one. All right, so this one, we're going to talk about eye color. So assume that brown eyes is dominant to blue eyes. And I didn't give you a letter to pick here, so we'll go with A's again, I guess. A father who is heterozygous brown eyes and heterozygous brown-eyed mother have children. What are the genotypes and phenotypes? So let's type in legend. We'll go with A again. So this combination, homozygous dominant or heterozygous, will give you brown eyes. And we'll go right below this, little a equals blue. So I'm going to type them over here. If we read our thing, heterozygous brown-eyed father and a heterozygous brown-eyed mother. So using what you know about heterozygous and their brown-eyed, they fall right here. So this parent, let me switch to my typing, is this combination, and this parent is that combination the same. So now what we're going to do is we're going to split the gametes. So one gamete's going this way, the other's going that way. Same thing. So type it in right here. Big A, little a, big A, little a. Now you're going to do the same thing you did before. Complete everything and bring out the Punnett square. These, if I'll try to make sure that it's black on there, you're never going to put anything in here. That's just an empty spot right here. One letter here, you should get the 2-letter combinations here. So let me type them in. So big A, big A, big A, little a, big A, little a, little a, little a. Okay. So I'm going to go to this side here. We'll write Geno right there. And like always, I like to start in this first square right here. So my first combination is big A, and I'm going to count how many I got. Big A, and I see I have, not A, I have 1. So go right here, check mark, I counted it. So now I'm going to go to my next empty square, which I'm going to use this one. So I see that's a big A, little a, the heterozygote, and I count 2. Go to my pencil, check, check. All right. And then I go to my last one, last square right there, which happens to be Boom, little a. Count it up, I have 1. I have to keep typing A, sorry. So now I'm going to go through and write up my genotypic ratio, which happens to be 1 to 2 to 1. So we can't reduce this down anymore. You know, that's just how it works out. So you'll put this, my advice is to always put the homozygous dominants first and then the heterozygous and the homozygous recessive last. It's just a habit that I have. If you have the right number combination, I understand the ratio you got. So that's your genotypic ratio. We got that. So 1 to 2 to 1. You add that up, it does equal 4. So let's do the phenotype. So what you can do is, I'm going to write pheno right here, make it a little bit easier for you. Write down the color, the eye color is going to be right next to it. So for this one right here, we know that big A gives you brown eyes. For the second one right here, we know that heterozygous gives you brown eyes. And then this bottom one gives you blue eyes. So I'm going to put my pencil right here. So what you're going to do for this one, the ones that have the like, you're going to combine. Now the reason you do that, because these are the same. Even though the genotype's the same, it still has the same phenotype. So when you type out your phenotypic ratio, you're going to add those together. Phenotypic ratio is going to be 3 to 1. All right, so pointer, boom, 3. Add this together, and you only get 1 of there. So 3, 1. 75% of the time, these people are going to have a brown-eyed kid. 25%, they're going to have that blue-eyed kid. So these, take time. You're going to do a few of them. If you're having trouble, please let me know. So let's talk a little bit in complicated some. Let me get a drink of water. Now we're going to do a dihybrid cross, and you only have one that you have to do on the homework assignment. It's probably one of the most tricky things you'll do, but don't worry, it's not too tricky. So what we're going to do is we're going to look at 2 traits. This one's looking at seed color and seed shape. So it's looking at yellow color is dominant to green and round is dominant to wrinkled. So when you go through that, and I'll show you how to separate the gametes, you're going to have a bigger Punnett square. The same comments are, concept is the same. We're going to split the gametes out, and then we're going to be bringing in the combinations. So in here, instead of having 2 letters, you're going to wind up having 4. It's the same process, just a little bit more work. Now, the one thing I think that really gets people is figuring out the gametes. So pretty much the same thing. Read your setup, write out your legend. We're going to figure out the genotypes of the parents. So we're doing 2 traits. So we're going to have 4 letters total. Remember in the previous ones, we only had 2 letters, but 2 traits. So 2 combinations. We're going to figure out the gamete combos, and we're going to do that via the FOIL technique. You probably heard of FOIL way back from math. So with FOIL, this is an example of a genotype we might have. You're going to have gamete combinations, and you're always going to have 4. Sometimes they might be some repetitiveness in there, but you're going to have 4 gametes always. You have to have that for your Punnett square. Now, how we figure it out, so this is allele 1, trait 1, trait 2 that we're looking at, sorry. So to figure out our combinations, we're going to do first, which is where we take the first of each of the different traits, the first allele, so big A, big B, there's combination 1. Out, so big A and little b, combination number 2, there's your O. Little a, big B, there's your I. And then last, little a, little b. And then we're going to take those and put them on the sides and the tops for the different parents, and then complete the Punnett square. And then we'll calculate our genotypic and phenotypic ratios. So let's do one. I know it looks scary, but we're going to get through it. So a lot of words here, but let's read through it. A couple is planning to have children. The male is heterozygous for tongue rolling and homozygous dominant for unattached earlobes. The female is homozygous recessive for tongue rolling and heterozygous for unattached earlobes. The couple is curious about how the possibility and probability of their offspring inheriting these traits. The ability to roll tongue is dominant over non-rolling. Unattached earlobes is dominant or attached. Complete the Punnett square and record the genotypic and phenotypic ratio. So always, let's do our legend. So we're looking at tongue rolling first. So we're using the letter R. So RR, and we're doing complete dominance. So tongue rolling is dominant to not rolling. So I'm going to put the first trait there, RR, non-rolling. Okay, here's your first trait. Second trait that we're looking at is, let me read, earlobes. So unattached earlobes is dominant to attached. So A, R, A, okay, little a, talking about the dominant trait, unattached. We'll go down, little a equals attached. All right. All right, so there's our legend. So now let's figure out the parental genotype. So then we have to read. All right, first one, dad. Dad is heterozygous tongue rolling. So we know that he's this. It tells us right there. And homozygous dominant for unattached. So keywords, heterozygous for tongue rolling, so he can roll. So that tells you right here he's this. And then he's homozygous dominant, okay, for unattached. So you know he's unattached, and then that homozygous tells you, boom, he's right here. So your first genotype for parent 1 is RRAA. That's the first parent. I'm going to put parent 2 over here. All right, the female is homozygous recessive for tongue rolling. Okay, so you know she's this. And heterozygous for unattached. So she's right there. So we're going to put the female a little bit further over here. So homozygous recessive, little r, and heterozygous for attached. Okay. So here's our parental genotypes. Remember, we're doing 2 traits. Here's trait 1, trait 2, trait 1, trait 2. Here's dad, here's mom. Now we're going to do FOIL. You need to get the gamma combinations. So this is where you're going to do the first, outside, inside, last. And you're going to do it for both parents. So we're going to get 4 combinations. Try to put some spaces between each of them. So for our first one, our first is RA. Space. And let's put a new period in between. We'll go with it. So our first, our outside is R, big A, okay? Our inside is little r, big A. And our last is little r, big A. Okay, so you see some of them are the same. That's okay. That's just what worked out. So first was the big R, big A. Outside, big R, big A. Inside, little r, big A. And last is big R, or little r, sorry, big A. Let's do moms real quick. So first is little r, big A. Outside is little r, little a. Inside is little r, big A. And last is little r, little a. Now what I want you to do is take one parent and move those 4 to the side, and for the other parent, move them over here. So I want you to take these 4, and each of them gets their own box on the dark blue right there. So I'm going to type them in right here. Big R, big A. Big R, big A. Sorry, big A. Little r, big A. So not an F. Little r, big A. This is kind of hard to do and collaborate. All right. So little r, big A. Up a little, RA, RA, and finally RA. Okay. So now what you're going to do is the same thing like you did before. You're going to bring the combinations together for each of the squares. Now every square is going to have 4 letters total because it's going to be a combination of these letters and these letters. Sometimes you'll see a lot of repetition happen. Like sometimes if I know it's always going to be the same, I'll draw lines down it, but I'm going to go through and type each of them in. Big R, little r, big A, big A. My advice is keep the R's and the A's together. It makes it a lot easier when it comes to the next step. And you'll understand when I get to it. And then my advice also is keep the uppercase first and then the uppercase, lowercase second. If you want to combine it, then rewrite it, you can do it, whatever works for you. This is where I really miss a whiteboard. This is a lot easier to do. And then I let y'all come up and do them too. Okay, little r, big A, little r, big A, little a, little r, big A, little r, 1, 2, oops, big A, little a, little r, big A, little r, big A, little a, r, a. Okay. Whew, I said these are the hardest ones. You only do one, and you know, don't worry if you don't really want to tackle it. I don't blame you. Okay, so now what we're going to do is do the genotypic and phenotypic ratio. So I'm going to, oh my God, I can't believe I just did that. I thought I was just going to erase the line. Crap, now I redo it. Sorry. Well, good practice for us. We should be able to get through this one a lot faster. So did not know the erase button erased everything. I'm so sorry, but this is good practice for us. Big R, big R, R, big R, little r equals rolling. Little r equals non-rolling. Well, now I know. So you can fast forward this until I get to the genotypic part if you want to in the video. I'm so sorry. I did not think that was going to happen. Welcome to learning how to use collaborate. Okay, little a equals attached. So I'm going to write the genotype closer to here for my parents. Let's see, dad was heterozygous for tongue rolling, big R, little r. I'm going to put dad on top for this one. And homozygous dominant for unattached. Boom. Mom is, boom, homozygous recessive for tongue rolling, little r. And she was heterozygous, boom, boom. So let me write in all my gamete combinations. The more you do these, the better you get. I believe I did that. It's the first time. Now I know not to do that in the future. Okay, so now I'm gonna type and fill all these in. Just can't believe that. Welcome to online teaching. Okay. A, big R, little r, big A, you're the same. Okay. So sorry that I did that. I really am. So now what we're going to do is we're going to do our genotypic and phenotypic ratio. So I'm not going to see erase button, but I'm going to type this over here. So we're going to have our geno right here. And my advice is to start with this first box right here. So this is your first combination. So I want you to find how many of them have this combination. So big R, little r, big A, big A. And then you're going to go through and count. So I see 1, 2, 3, 4. So I'm going to write it over here. Big R, big A equals 4. And just like before, I like to check them off. So I counted this one, I counted this one, I counted this one, and I counted this one. All right. Go your next empty one. So if you want to go across or up and down, it's up to you. Little r, big A, big A. So count. I count 1, 2, 3, 4. So we're going to type it down right over here. Okay. And that one equals 4. All right, so just like before, you could cross it out however you want to do it. Check them off. Boom. All right, those. Go to your next empty one. Big R, big A, little a. And I count 1, 2, 3, 4. So right here, big R, big A, little a. I count 4. Cross out. Boom, 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 boom. And then I go to my last one, which happens to be little r, big A, little a. And then I count 4. Now, one way that you can check your math to make sure you counted everything, that should be 16. Sorry for that. Check them off. I know I counted them. That's one way to check. Everything works out right here. So now let's go through and write the genotype right next to it. So if it types over that, I'm so sorry. So what we're going to do, we're going to write the genotype for this guy right here. So go back to your legend. So this top one, they are going to be rolling and unattached. Yeah, it's going to overlap a little, sorry. Your next one is going to be non-rolling. And they are unattached. Right below it, the next one, so they are a rolling, unattached. And then our final little guys here at the bottom, they are non-rolling, unattached. Okay. So now what you're going to do is match like with like. So from what I see, you've got half of them that fall in this category and half that fall in this category. So let's type out our ratios. So our genotypic ratio is 4 to 4 to 4 to 4. Or you can reduce it down to 1 to 1 to 1 to 1. And our phenotypic ratio is, add them up together, 8 to 8. Or you can reduce it down to 1 to 1. Okay, so hopefully after that little snafu that I just did, I'm so sorry about that, you kind of go through the steps. So what you did was you wrote out your legend, you figured out your parental genotypes, and then you did the FOIL to get the gametes, combined them all into this Punnett square, computed your genotypic ratios right here, and then take the genotype and determine the phenotype, and then get your ratios. So like I said, you're going to be doing this on homework. If you need help, let me know. And practice, practice, practice. So I'm going to stop this one right there.